Right. Hello, everybody. Thank you for joining the ChUG podcast. My name is David Leclerc. I'm the host. I'm here today with Daniel Vassell, who runs everything flow stometry at MSU, and Cezary Shizhevsky. I practiced 5 minutes. Shizhevsky, who runs the HDID core facility at the University of Chicago. So it's the Human Disease and Immune Discovery group. And today we are talking about spatial biology. We all survived the solar eclipse. This is April 11th, and nobody got hurt. Which I thought the eclipse would would do more damage than that. There's a bunch of sinners that I thought would actually get their coming, but yeah, there's a bunch of gods here that are just slacking. Thanks for coming, guys.
Yeah, anytime.
So spatial biology is on my mind because Danielle at some point said that it's going to replace flow stometry, and all my life's work will be made useless. And so what we're gonna do is going to start discussing what's the place of spatial biology and how it's going to crush flow stometrists, and then we'll go over what's available on the market, and finally talk about how spatial biology looks like in a core facility. I myself have not done any research. I don't know anything about spatial biology, so I'm going to rely heavily on Danielle and Cezary to make this entertaining. So again, thank you guys for coming.
So to be fair, I don't think it'll replace flow cytometry, but I think that it can replace quite a few instruments, and it definitely has a place in flow cores.
Right. You would put it straight in the flow core and not its own thing?
Yeah. Although my experience is not directly with the spatial biology, but with like high content imaging, high content screening. And in that case, I really do think it has a place in the core if the decision is flow cytometry core or microscopy core. And again, I think that the way that flow cytometrists approach data, we're just used to much larger data sets. And so we kind of have that background in bioinformatics that's really already there for processing, like these just incredibly large data sets. So I'm not too familiar with like the systems that are all using like a wide field scope and, are doing the automated antibody washings and rinsings, those types of instruments, but just purely the high content. And the high content is different in and of itself. It's imaging, and for those that aren't too familiar, so there's the spinning disc confocal that was an upgrade to confocal microscopy that I think came out in 2019, 2020. And if you have like a food processor, and you can imagine like, if you put the cheese in and it kind of grates the cheese, that's kind of what the spinning disc confocal is, except for every little hole is a pinhole. And it spins really, really fast. And so it can take confocal images at roughly 1,000 times faster than you can with a normal confocal. Well, all of a sudden then we're taking images fast enough that it's kind of almost at the same level as cytometry in terms of, acquisition. And so a lot of these instruments will say that they can, process a 96 well plate in, 4 minutes or less. So that becomes pretty darn fast pretty quickly. Then you throw on a lot of the software, the analysis software that exists in the field of microscopy. And if those of you that are unfamiliar, it's come a very, very long way, especially with a lot of the AI features and machine learning where they can find cells pretty darn easily. It's just boom, finds them, segments them. And then a lot of these instruments too will just automatically export it to an FCS file. And so if you consider it, that placement in a core, you have an instrument that doesn't require a user to subculture their cells. They can just take the 24 well plate, the 96 well plate, boom, put it in the machine. No eyepieces, any of that anymore. Auto focuses, auto finds the wells, goes, reads the whole plate in 4 minutes, and then it'll export you an FCS file. Now you have an instrument that doesn't have a fluidic system, is incredibly easy, cheap to maintain and run. So that I think has a place in cores.
Is that like, what's the limitation there? Like how many lasers, detectors, that type of stuff? How many markers would you actually look at for?
I would say it's the same limitations that you could reasonably have with flow cytometry. You can always add more lasers. It's just if you want to pay for that. The difference in microscopy compared to cytometry is, imagine you have your 488 laser and then your detector set off of that. We're used to having multiple detectors per laser, where theirs is generally one detector sort of per laser. That's a generality. There are spectral systems, there are white light systems, but generally you can think of it as like a rotating filter wheel. So if you wanted to have 12 colors, you could. It'll just be one laser, one filter, then the filter wheel rotates, next filter, go over to the next laser, and so forth.
Okay.
Yeah, I mean, I think truly, right, technically looking at it, it's really just one detector because it's a camera detector, right? What we do, we put a different filter in front of it, and then we acquire different spectral part of light that we obtain from the fluorescence modality, for example. But yeah, technical limit, I don't know about microscopy that much, but I think 8 colors is something that we see on the higher end that routinely used right now. I think Leica, the white light LED system and so on. So that's about that much. But then becomes a new kind of advancement in a multiplexing, right? How do we get more and more and more and more? And there are many different techniques that allow us to achieve the multiplexing there, right? So we can have images that are generated and stained really, really well with single cell resolution, 10, 20, 30, 40 or more protein, targeted by different antibodies, for example.
Mm-hmm.
On the spatial transcriptomic side, you have thousands. Really, right now, we do have technologies with ability to detect and quantify thousands of different transcripts at single cell resolution. Hundreds, maybe, right now, standard, but few kits released by NanoString, they claim to do 6,000 right now, right? So it's advancing very, very fast, very rapidly, and I think it's a really fun field to be in and observe a lot of different pieces moving through that whole process of discovery from the very beginning, image acquisition, through image processing, to the analysis, which is an open box right now, in my opinion. Like it's not that easy. Daniel just said it's very easy to segment cells, but if you get into nitty-gritty details, actually, like it's not that simple, and there's so many different methods and algorithms that are trained on different data sets that allow you to do it one way or the other and achieve better results at some point.
I think that's where I'm getting lost quickly. Like it sounds to me like there's a bunch of them that relies on actual flow force signals, and then you kind of jump to other platforms, and now suddenly we're talking about
I
sequencing.
think it's confusing.
Yeah,
it's not because it's such a new field and it's so much going on. Everything is brewing really in that field right now. The new companies are coming in, and then some of them they seems to be disappearing and new technology. But for me, it was always easier to kind of navigate through that field by subdividing, right? So like in my head, I always subdivide first at the very beginning, proteomics from transcriptomics. Or we can even say, not really transcriptomics, it could be spatial genomics, because there is more going on than just the transcript detection and transcript quantification. So, but for me, it's just first step to separate different techniques, right? And different approaches. But then you can subdivide all those two big fields into many different categories. So, for example, if we look at the modality, what do we use to detect proteins, right? There are at least a few different approaches. One would be mass spectrometry, right? Like mass spectrometry was actually maybe not the first one, but very first at the beginning that was commercialized. And our friends from Fluidigm, right?
Yep.
Fluidigm, right? They did that at the very beginning. And it's not Fluidigm anymore, by the way. It's
keep forgetting Standard BioTools.
Yeah, Standard BioTools, right? And basically what they did, they repurposed the CyTOF or Helios or whatever. And now you can look at the slide and stain this slide with, metal conjugated antibodies and ablate it with laser quite well, at relatively high resolution and detect everything. The beauty of this, the main advantage, you do everything in one pass. So you can stain with 40 antibodies, right? run the laser over that tissue just once, and then quantify all those different metals conjugated to antibodies and detect your whatever you want to detect, right? That's really nice. On the other hand, you have fluorescence as the main modality. Then, like Daniel said, you're detecting some kind of colors, like in a flow cytometry world. But because that limitation of the sensor, detector, and so on, right, you cannot combine all 30, 40 antibodies and look at them at the same time. We can do that with Aurora, right? We can do that with advanced instruments when you have 30, 40 detectors from different companies. But in terms of microscopy, you are limited to maybe 4, maybe 5, maybe 8, as we said, right? So then what do we do to detect 40? We go in cycles. So we detect 4, remove the signal, add reagents that we need to add to detect another set of 4. And that iterative aspect allows us to look at 10, 20, 40, and hundreds of antibodies, really, if we need to, right? So that's another modality. I kind of like the combination because there is a spectral instrument and a company that allow us to use the spectral aspect, spectral mixing to get better idea what do we see if we combine a lot of different fluorochromes, but they are different, overlapping, but different enough. And I think the instrument is called Orion, right? From one of the companies, it's commercially available. And then I think limitation there, the highest plex I was able to find was 17 antibodies, right? But they were able to resolve 17 antibodies using spectral mixing on the tissue, again, single cell resolution. Pretty cool, right? And then there is the old method, right? When we use just standard type of immunofluorescence approach, but it's a cyclical immunofluorescence. So you add primary antibody, you add secondary antibody, take an image, right? And then you are limited to maybe 2 antibodies at the same time because, rabbit versus chicken versus donkey and whatever you have there, right? So you have to be careful how you combine the primary and secondary. Then you strip the primary antibody, add another set of 1 or 2 primary antibodies, come with the secondary, and you do that over and over. And people have been doing that for a number of years already, kind of manually, right? Like you do it, you put it on the microscope, you stay with 2 antibodies or 3 antibodies, put it on the microscope, take an image, takes a whole day, take it off the microscope, use the same sample, stain it again with new 2, 3 antibodies, and do it over and over and over again. And it takes a week or 2, but then you have 40 plex.
Right.
Right now, it's kind of automated. There are some companies and some instruments that allow you to do it in an automated kind of walk-away way, application, and then you have that instrument doing the fluidic part for you.
So that's
a little bit more complex.
That's a good point. So it sounds like I've seen spatial biology as a fairly recent thing. Like the first time I was asked if I was interested is when we were thinking about what else can we do with our mass cytometer, and we look at the Hyperion for half a second. But it sounds like that's been going on before the Hyperion. Can you set a date on like how long have people doing that type of applications? No, you don't have to.
I don't know, right? Like I would have to go back and look at that first original paper of the cyclical immunofluorescence. I think that was one of the first approaches. I don't remember when was it, but
it's at least 15 years plus old, though.
Right.
And I guess it's hard to say what's really. I mean, it's a natural progression in the field, right? Because what's spatial biology? It's ability to observe cells in their natural kind of organization within the tissue, right? That would be the simplest definition. So if you use that definition, the typical microscopy that allows you to see a couple different cell types, right? That could be a spatial biology. But then people were, eager to add more and more and more information, or to obtain more and more information from that same tissue, right? So they're coming up with different approaches to do that.
And so going back to then the hardware side, because I think that was a great overview on the assay side of things and what people and folks can do. I think you have the breakdown for, again, the systems that have optimized like image quality or image acquisition speed, and you have the systems that have optimized I would say like the assay design. So just like you said, the instruments that will automatically do the stripping, right? You can load up your 20, 40 antibodies and they will automatically take the sides, image them with 4. They'll automatically wash them, strip them, add the next 4, and they'll just keep going in cycles like that. Most of those are using like a wide field system. So that's a wide field is, what you probably have is like a benchtop microscope. And I've seen a lot of those are using more digital techniques to make the images look better instead of like, hardware techniques. So that's processing as much information as easily as possible. And I would say that the flip side of that, the high content bit, is processing simpler things as fast as possible. And I think that it's hard coming in with what you think you understand about like what an image needs to be. And, when you really need that resolution, you can spend the time to see all those specific minute details for, I would say, what a lot of the spatial biology is. Well, high content is in that space. It's a different animal entirely. So in the spatial biology instruments that I've seen, right, you can put your slides in, you can click it, you can hit go, and again, you'll spend a lot of that time in the analysis as well. Like when you're doing those 40, 50 colors and stuff. For the high content, most folks are only looking at a couple of colors. And the idea is like we're running hundreds of plates a day. That's the goal for sort of high content, which is why that sounds a lot like flow, right? I want to run 100 samples. I don't need a lot. And then, think about your average user in the core. The average user isn't doing a 50 color panel. It's limited application. Most folks are doing 1 or 2 colors. And that's what I think is really interesting about these instruments in a core is, again, if it can do a 96 well plate in 4 minutes and they don't have to subculture their cells, I mean, that's an incredible amount of time and savings. And again, like they only need 1 or 2 colors. If you're just trying to figure out, as a screening tool for CRISPR, are my cells green? Are they red? Are they this? Are they that? It can do that very quickly and very, very simply. I would say that that's then your entry level. So to get the average data that our average user is generating, you have an instrument that is potentially faster. And when we demoed all these instruments, we actually did a head-to-head with that, with a lab that does regular screening, right? A CRISPR screen, and it either expresses GFP or TD tomato, and started the clock for when they started processing their samples, pulled them out of the incubator for flow, and going right to the high content. Data was the exact same, right? When they processed it from high content and got their FCS file, made their plots. But, they cut out about 2 hours of the sample processing time that they didn't have to do because they could just take it out of the incubator, boom, put it in image. When they were done, they could put it back. So now it also allowed them to do time with the exact same sample set. Or for a screening assay, you don't have to take half of the well, save it in case you want to perpetuate that, that well. They could just screen it and keep going. And then even the runtime to go through, their 24 well plate in flow to run multiple samples with an auto sampler to get those, 10,000 images. It was about the same time as running about the 2 or 3 samples and trying to collect a similar number of cells to get about like 5,000 cells on the imaging system. But they don't have to look great, right? You can use a 10x, you can use a 20x. You just need to know where the circle is. And it's the same thing. You're just quantifying the total cellular intensity. And so I'd say like that is your entry point for these instruments into a core. And I would say that it's just like talking to somebody that's never done flow. It's hard to come at them and say, well, I hear you just want to quantify GFP, but have you considered doing 50 colors? And it's like, whoosh,
Right.
It's like, you can work there and you can get them used to it as well. And that's where I kind of see trying to get cores used to these instruments because they can do a lot of the exact same things that we can do. And it's all of the hurdles that a lot of cytometrists have had with imaging and microscopy in the past aren't really there anymore. That's like the autofocus, the auto laser. I've talked to a lot of folks that, either have an astigmatism or something. And they're like, the whole reason that I did flow is because I could never get things in focus on the microscope. And it's like, you don't need to anymore. Like you click the button, boop, boop, boop. Boom, it locks into the position, it goes and it reads. And so when you come out with how simple these things are nowadays, now you can start getting users interested. And that's what you're asking about in terms of growing the core and growing the use of these instruments. They're just that simple to use that I can train people that have never done microscopy before. Here are the 3 buttons you click. Done. Yeah. It's just that simple and that easy. And then again, as a running a core perspective, most microscopers don't have service contracts on these things, because when you need to replace something, the community is great. You can add a laser, you can call people up and how to fix and repair these things. They're also just incredibly robust. I mean, think about whenever you have a service call, it's usually related to the fluidics, not necessarily a laser going down or needs to be aligned or adjusted. And even the laser alignment adjustment, it's just usually a single mirror, and it's just, not hard to go in there and tweak that. So that's the entry point. Then you start getting people interested. Then you go, hey, you know what? You have a few minutes left in your reservation. Let's switch over to that 40x, that 60x. Oh, look, there is, subcellular localization in between some of these different samples here. Let me show you how to build out an algorithm to go ahead and process that. Or again, with the machine learning algorithms on these things, it's so easy. You just go, let's just draw circles around these couple weird looking cells. The software then figures out how to identify those. And then let's look at those in the rest of your sample. And just like what we do with flow already, where, hey, you tried one or two things, but hey, I just got some samples of new stuff in my fridge. Let's try spiking that in. We can see if ROS matters or something. Once you start getting that interest, then you can open up the floodgates and go, now we can start doing 50, now we can really start looking at spatial biology. What is the relationship between, these cells, their transfection efficiencies, what kind of interactions are occurring? And again, I think that it's so important for us as flow cytometrists to have this conversation, because this is an instrument right now that could go in a flow core and it could go in a microscopy core. And I think ultimately it's something that's going to bring us both together.
Yeah.
Right.
I agree. And, like what caught my attention when you said the processing time, right? Like it's so much different. I come from a little bit different perspective, because like I'm all about human studies and human-based studies. And for me, using flow cytometry for a couple decades, right? Like the difficult part was always, how do we get these samples to the lab in the most efficient way, right? Timely, make sure they're all kind of treated the same way, right? And you have a clinical trial, and you're getting, samples at different times and so on. And then you have to do the dissociation, right? Like, how do you dissociate the cells? What kind of enzymatic cocktail do you use? What are you missing? What's coming off that sample perfectly, and what's left behind and clumped together and dead? That was always my concern, right? Like, how do we make sure those samples are really the same and equal and represent the tissue that we are supposed to be looking and comparing? Right now, with the spatial technologies, what we often do and we often work with are those paraffin embedded formalin fixed samples, right? Which is the standard, gold standard in the clinic, in a pathology, right? They take the piece of tissue, they dunk it in a formalin, and they leave it there for a few hours, for a few days, and then put it in a paraffin block. And it's stable for decades. We run samples that were 30, 40 years old, and they look amazing, right, on the protein. So that's, for me, the beauty of these approaches right now. I can go back, I can get the samples that were collected days, weeks, and years in the past, right? And then analyze them all together at the same time. And, be kind of sure that I'm not losing any cells there, because whatever I can see, it's really there, right? It's there in a tissue from the moment that that tissue was removed from the patient. That's amazing for me.
Are there any applications currently done in flow that spatial biology could take over? And I'm thinking we're currently trying to build a pipeline to do multiplexing assays with the robotic core here, the cell screening core facility. They would prepare the plates, and then we'd run them on our instruments. The instrument, each plate is about 45 minutes. If I threw my plate of beads on a spinning confocal, would that allow you to build the same kind of standard curve and make the same kind of readings, or am I not understanding how this thing works at this point?
You are. So if you think about when you go to load your beads on the cytometer,
which one I'm not understanding, or
no, you're good. But it can.
Okay.
If you go to throw your beads on the cytometer, right, there is a fine point for every detector where the voltage is optimized and your positive or negative populations are resolved.
Yeah.
But past that, if you start increasing or decreasing your voltage, they're moving together, right? There's reasonably no difference. And, when you draw your gates, it's still 50%, 50%. The equivalent to that is in the microscopy is like looking at your gain and exposure times, but there's always a background, and then there's your signal. The longer that you do your exposure time for, the greater you turn up your lasers, you're going to get a bigger difference between background, and your true signal. So there's a minimum time where like that is all that I need, that I can go a very, very short interval. Again, if I'm only looking at 50%, it doesn't matter if they're this far apart or if they're this far apart. So it's up to like what you could need, but you could reasonably try to find something that's just as bright. I think Slingshot like has new beads now that have like different levels of expression for your antibody binding. So you could try to find something similar and to match it. But overall, like when you get one that's close enough and you're like, that is good enough resolution, like it's pretty good across. Or you do it once and you go, here are the exceptions, just like you do in flow. This is how I need to adapt and change it the next time that I'm going to go. But I really do think like if you were going to do a massive study like that and you kind of knew roughly where things were at, you could find a reasonable, laser power exposure time that would be applicable across all things.
All right. You guys want to talk instruments?
Sure.
How do you want to do this? Like start simple and go complex, or by date of birth of each platform, or?
I don't know. I mean, for me, there are microscopy-based application, but there are also non-microscopy-based application to kind of look at the spatial biology. And then on the microscopy side, you have a very low resolution, some kind of low pass of the tissue, for example, right? And then you have a really high resolution objectives where you look at subcellular, interaction between molecules and things like that. So again, could be very simple, it could be very complex. But I think maybe, like there is the spatial transcriptomics field that's not microscopy based, that's kind of sometimes, omitted from the conversation because, it's not microscopy based anymore. It's more sequencing. So, going back to the course and, different course and different involvement in this new technology, I would add sequencing core because that's sequencing based type of analysis, right? And very simple on the instrument side, when you get the data, the spatial information there, because what you do, you kind of put, like in terms of the instrument, you have basically two different instruments on the market right now. There's one from 10x Genomics, which is the Visium, right? And then you have one from NanoString, which is the GeoMax. So what you do actually, like you simplifying, you take all the RNAs from the location of the tissue, right? And then you sequence that on the Illumina sequencer. And then because you have a way to go back to that location, there's a spatial barcode, right? Then you can realign that sequencing information back to the location. And then you can say, well, this is the expression we have per unit of area, right? Kind of. So in terms of the Visium instrument, basically you have a special slide with bunch of little spots printed on it with those unique special barcodes and something that will grab your RNA from the tissue. In case of the NanoString instrument, you draw your area as you like, right? And then you take all the transcript from that area and you sequence that, right? So that there is no microscopy, but you still get the spatial information. The problem here is resolution, because the resolution is only as good as your size of the spot in terms of the Visium, right? Or the area that you draw. The bigger the area, the lower the resolution, because that's the transcript information quantification that you're getting from, right? So going back, but those are amazing techniques actually, right? Like they work extremely well. And what great about those is that those are unbiased in a way, right? So you sequence everything that you can grab. You can have 20,000 genes. Illumina sequencing been on the market for a long time, and the sensitivity is pretty good, and you detect it a lot. You don't have to design your panel. You don't have to design your panel of antibodies, or you don't have to design your panel of, hybridization prop to look at only set of the genes or set of the proteins. So that's kind of, for me, the entry level could be also there with the help from the sequencing, genomic sequencing course, right?
Right. My understanding with the sequencing part is it's going to be very expensive to run your samples. It's a bit like 10x genomics and flow stoichiometry, but then the difference is the amount of data is just flabbergasting. Is you get so much more than is that pretty much the same idea with sequencing versus like purely.
Yeah, that's very true. I think I never did like cost analysis for the Visium, but I think it's about $4,000 for a slide, right?
Right.
I don't know if it's the sequencing cost is included, but like I think it is, right? So about $4,000 for a Visium slide. I don't remember the size of the area. Maybe it's somewhere around 8 millimeters, right? Which is substantial. That's quite a lot. But yeah, it's expensive. I mean, everything new is kind of expensive, like. By the way, I kind of going back to comparison. I always compare in my head the spatial kind of techniques to flow cytometry because that's what I really know and what I really like. And when we're talking, I just realized actually, like I was always pushing Aurora panels, right? Like I wanted to have the biggest panel on campus or on the block on Aurora. So I was able to have maybe 42 antibodies, like, my best ever highest plex panel on Aurora was 42. And I did this as a proof of concept, and like none of the users actually ran that because I think the maximum was close to 30, right? Like from the users in the core. Well, I realized just now that the panel using our spatial biology, system, proteomics, and so on, it's I think 54.
Ooh.
54 antibodies.
Yeah.
And it looks great.
Yeah.
Right.
That's the thing. I mean, there's always going to be limits when you come back to fluorescence, right? Like just the algorithms that are used for unmixing and the inherent noise that continues to accumulate that just isn't present there with other systems and the ways that things work. And that's like the field of cytometry is going to have to pivot at some point too, because no matter how many lasers we add, there's only so many fluorophores, and like having to rely on spectral mixing. I mean, the field of microscopy has gone past that with fluorescence like lifetime imaging and trying to use other techniques in order to separate different things out. And we'll have to do the same, whether that's adding more lasers, again, going into the deep UV, whether it's trying to come up with more fluorochromes, but how do we keep increasing the number of things that we're going to be adding?
Yeah.
I mean, I think on the protein side, ultimately we are limited by the number of good antibodies that we can use, right? Like we've shown that, and I think it was Akoya who showed the staining of the tissue with 100+ antibodies, right? And it seems to work just fine. There seems to be no limit on the technical side of the instrument. It's just the limit is how many antibodies we can really have and use that works in this type of assay. So, a couple hundred. But I think the limit on the transcriptomic side, this is just the number of transcript we want to and can detect, and that's it. Like that, really.
Yeah,
and the cost, of course, right? The more you want to detect, the more expensive it is.
Yeah.
Interesting too, because I know there's kind of been a resurgence within like Q dots and making really, really small, really, really bright things. Because in the same way that like the Visium works, right after that initial staining, you could almost run that inflow to increase like your throughput if you were able to kind of correlate it back. It's just interesting. It's interesting as like the three fields that were always so diverse are coming together, starting to have significant overlap with each other. I don't know. I think it's exciting.
Going back to this coming all together from different fields, actually, we may want to talk a little bit about analysis because that's a completely different beast. But in my opinion, field is moving into integrating different aspects, different type of technology and different readouts, and combine them all together and get as much information as possible. It's not really additive, right? So what I mean by that, there are algorithms being developed and used right now that allow you to look at different modalities and different type of technologies that were used to analyze the same sample, right? So let's say you run single-cell RNA sequencing, right, with dissociation and so on. And then you also run flow cytometry experiments on the same sample. And then you take the tissue from the same location, the same patient, the same type of tissue, and you run it using any of the spatial biology application. Could be spatial transcriptomics, could be spatial proteomics. Now we can, or we will be able very soon, I believe, combine all those data sets and analyze them together, right? So that's the conversion again, coming on the analysis side. It's kind of amazing because, again, not additive really, right? Like you have different pieces of information coming all together, creating this amazing, huge data set that I don't think we understand yet how to really dig through and analyze correctly, but I think it's coming.
So we dropped a few names, and I have to check as we walk through. The Visium is the 10x Genomics. When we talk about the Akoya platform, I see a number of them, and they are mostly image fluorescent based, or do they do a mix of sequencing and
So Akoya, right? Like I think it was one of the first company. You know what? Actually, like I also did a little bit of reading a couple of days ago, and I can say that a lot of these approaches and companies come from one single lab, and that's
really?
this Gary Nolan lab from Stanford, right? Because Fluidigm, it's Gary Nolan.
Yeah.
Ion Path, it's Gary Nolan.
Yeah.
Koya is actually Gary Nolan as well, right? So like it's very prolific. Amazing, right?
Yeah.
Crazy. So, we talk about ion path a little bit, the mass spec approach. And then there is Fluidigm, right? Which is also a mass spec approach. Akoya is cyclical, right? So you run the experiment, and I'm partial because I do have a Akoya system. It's the first generation, but we couple it actually with the spinning disk confocal microscope for, because we wanted to have nice images. But there you cycle barcodes with different fluorochromes, right? And they're corresponding to a bunch of antibodies that you stain your tissue. So it's a cyclical, but you don't cycle antibodies. You cycle the secondary reagent in a way. And then, Orion is the spectral, right? From this company, Rare-Site, I think. I don't have any experience with it, but it looks kind of like it looks really cool because it reminds me the revolution that happened when Aurora was released, right? By Cytec. And then there is, for example, Lunaphore. This is a technology which cycles the antibodies, right? So and automated. So the instrument basically adds primary antibody, adds the secondary antibody, and takes the image, automatically, and then removes the signal, add a new set of antibodies, primary and secondary, and so on. And I think there's a system from Miltenyi called Maxima.
Yep.
The same kind of application, right? Cycling of the antibodies. I looked into Maxima a long time ago when I was kind of picking different instruments as a possible one for the core. And we went with Codex back then. So Codex is the original name from Akoya, but then rebranded this, and it's called PhenoCycler right now. And then they have a low number of markers that can be run more on the clinical side, traditionally maybe 812. And I think that's Pheno something, I forget the name. But Maxima, was back then 3 years ago, was really, really expensive, and there was not much of the independent data back then. I don't know how they are doing right now, but it's kind of similar. And on the spatial transcriptomics side, I'm aware of 3 different companies with mature commercial products. That would be the Visgen, which has the Merscope, right? Then there is 10x Genomic with their system called Xenium, that's a single cell resolution, or the Visium, that's the spot-based, right? Not a single cell resolution and sequencing based. And then you have a NanoString, that's again 2 different instruments. One, this is the lower resolution sequencing based, which is called GeoMax. And then the new one is CosMx, right? That's a single cell resolution And here we're using like a hybridization probes that just bind to the different transcripts. So you are designing, or your company has a panel for you that you just add on your tissue, and then you can detect and quantify different set of genes, right? And then that plex varies between different companies, so a few hundred to a few thousand, between different applications. It's kind of really cool, right? And I think Merscope was the first one in a single cell resolution market. And interestingly, right, like both two companies, the 10x Genomic got their license from a George Church Lab company, spin-off company from Harvard University, right?
Mm-hmm.
And then Merscope was based on the Marefish technology from another lab at Harvard, right? So like, again, kind of very prolific institutions spitting up very new, exciting technologies for us.
That's awesome. I didn't realize the codex was Akoya, and I didn't realize everything came from Stanford.
Right? I know, right?
Do you know if Ian Pat is still around?
Who? Sorry?
Ian Pat?
Yeah, they are around.
They are?
Yeah.
I thought with Akoya, I thought they would just said, okay, we're giving up on the mass cytometry stuff, and
yeah, they have a system. I forget the name. I think it's like
I think it's called the
AI scope something like
okay.
like it's yeah. They're around.
Right.
Yep. And again, I think like that kind of reminds me again, cyto versus spectral versus many different detectors type of approaches in the flow cytometry field, right? Like the metal conjugated antibodies were very, very popular for a very short time, right? Like we all jumped on it. We wanted to run 20, 30, 40 plex panels, and we did. And then cyto came around, and we went back to fluorochromos.
It was just a matter of like the cost of the data point. I think was what drove the decisions, because
I mean, different workflow a little bit, right? Like I did a couple experiments using using cyto, and then but I quickly realized like the staining is a little bit different. You have to be careful of the contamination, and like I think the first time I did, I autoclaved something and I used it in my essay, and that light up. I forget what kind of metal is in a shell of the micro autoclave, right? That just gets into the reagent. So like you have to be careful with that. I think it would be very successful if Cytec, didn't come up with Aurora.
Yeah.
Right.
Probably.
We'll be still doing running things on Cytec, I believe. But yeah, that's a summary of the instruments.
How do you compare them now? So we have the sequencing stuff, we have the mass cytometry, and we have the fluorescent based. Let's stick to fluorescent based, I guess, because like that's gonna be comparing things that kind of work the same way. How would you compare? Which is best for you? What are you looking for?
I think it's hard.
Yeah.
Well, if you want the best instrument, right? You pick random company from whatever list you want to generate. You call the first sales rep on the list, and they will convince you their instrument is the best. So that's one way to do it, right?
That's
because
easy.
that clearly is the case. Like I looked into a number of different instruments for fun and for really identifying one that I want to have. And that was my understanding. That's what's happening, right? Like there's little independent information right now still because it's so new, right?
Yeah.
But you can start looking at publications, and there are a few just coming out, and then you can make your own decision, right?
Or is it also that they kind of work different, or I'm trying to and again I don't really know. Is it that they kind of are optimized for slightly different type of results? That they would say, oh, it's better for this or that, or not really?
I don't know. I don't really think so. I think those companies are trying to create the best possible product that they can. And then I think it's our job to really find which one works best, right? Like I don't think they are in the business of directly comparing. They are in the business of making what they can as the best and then advertising this as the best. So it's not that easy. I think, many different aspects to consider: cost, right? Ease of use, workflow that needs to be applied on those samples before the image acquisition, for example, right? They're all a little bit different. So you have to do your research and really see what will work for you, right? Resolution, again, something to consider. Do you just want to look at single cell, or do you want to look into subcellular, composition and kind of detect? I don't think there is a single answer. This is the best instrument. It kind of depends, but they are kind of similar on the paper. I don't know, right? Like I don't know how to. I have my favorites, right? But I may be wrong, so I don't want to say anything.
Also, it was similar and different in a lot of ways for the high content stuff. So last summer we did a head-to-head with 5 different high content imagers, and we brought them in and did demos. Every company seemed to have one little aspect that was different, and especially you can put this to the sales folks. Well, why you over other people, right? And they, these 1 or 2 things, but it's hard for a core, given the diversity among our users and projects, to say, but which one of these is most important for my user base for incorporation into a core. And so we demoed a number of instruments. So we did the Thermo Fisher's CX7 LZR Pro. We did Zeiss's Cell Discoverer 7, all the way up to their Aries scanning and their spectral module. We did Biotech, which is now known by Agilent, their Citation C10 instrument, along with the addition of their BioSpa, which is a robotic incubator. We did the Molecular Devices Confocal HTAI, and we did Yokogawa's CQ1 were the 5 that we did, and they all seem to excel in their own right. Like the thermal instrument had a far red laser that nobody else had. The Zeiss Cell Discoverer wasn't necessarily as fast because it's not a spinning disk confocal, but they have an Aries scanning module that allows them to go much faster, and theirs was the only what had native spectral built in. The confocal HI was supposed to be a really nice middle of the road, and we talked to several cores that had it, but due to scheduling things that fell through, we actually weren't able to actually demo the physical instrument. And the Biotech Citation C10 was really nice for user friendliness, as well as its price point. They use the same Gen 5 software that people use on their plate readers, they use on everything. So that was super easy for our users to pick up. It was also incredibly easy for if you want a core, you get an instrument that people just want to click a button, and do, I want to click 2 buttons, I want to get my data. It was really, really great for that. The Yokogawa CQ1 so they're the ones that came up with the spinning disk technology. Everybody else licenses it from Yokogawa. But they also make the instruments too. I would say that they don't have anywhere near the marketing budget that everybody else does. And we didn't even have them on our list until it was recommended that we look into them. But they have another invention called the micro lenses, which kind of throws out what we think of in terms of resolution with numerical aperture, because it's focusing more light to the objective. So it's you're starting with more light in the first place. For us, comparing across all of these, we wanted to have a variety of applications that it could support, but ultimately it came down to speed. And so in our head-to-head, the Yokogawa instrument was about 10 times faster as everybody else because they were collecting so much more light to get the same resolution. We could go 10 times faster. We didn't need nearly as much exposure. But when we compared it, we also looked at our users. We had groups who wanted to do spheroids. We had groups that wanted to do plants, things that seed rehydration, all the way down to trying to find bacteria within a live host. A whole range. We had groups that had been doing this, so we knew that their stuff, we already knew that it looked really great. And then we even picked some users that were negative control, I'll say, to see, you know, how well can this do with a truly wide range of sample quality, and tested them among all the instruments. And I'll say that they all can get the job done. So in that regards, like every company is really trying to make the best instrument that they can. It just comes out to little differences that are going to be what is best for your user base. If I had the money to buy 3 instruments, I don't think I would have bought the same instrument 3 times, right? I definitely would have gotten ones because they all excel at slightly different things. But the thing we're not supposed to ever talk about, which is money, is where I was shocked. So I would say that at the entry level among all those different instruments, you could probably get in for about 175,000. And then if you really,
that's for the instrument itself.
Yep, that's the instrument with all the lasers, the control modules, the computer, the software, and everything.
That's cheap.
Yeah. And again, if that's the same price as a cytometer and you don't have a fluidics and stuff to deal with, and it's just as fast, that's why I keep saying, like, we need to start thinking about these things because they certainly have a place. And then you could go all the way up to, $1 or $2 million, if you will, or wanted something with every single bell and whistle and feature, possible on that. It's interesting. The other one that I didn't get to mention is there's a growing interest in morphological parameters, right, within flow. And if, you necessarily can't have an instrument there, you can get that same morphological instrument from this. So you can take these instruments, which is the same lasers, the same filters that we have in our cytometers. You can port existing panels right over with the added benefit now that you don't have to subculture your cells. And it's all the data is right there. You have the images. So as these algorithms get better and better with, morphological parameters and what they mean, you've got the data. It's almost, like saving your tissue samples that you can come back and look at them another time. And if you want to, you could fix them and come back and look at them a different time as well. The other benefit is the biosafety. So at MSU on our confocal microscopy core, they're not allowed to run BSL2 samples, but we see a lot of interest in growing among BSL2 samples, BSL2 plus, with a lot of kind of extreme pathogens. And what's nice with this unit is that without a fluidic system, there's no risk of aerosolization.
Mm-hmm.
And additionally, we've done some studies with different types of tape. So you can actually tape up the well plate, the top and the bottom, sealed on the side so that it allows gas exchange through, but it keeps the unit sterile so that even if a person drops it, like the liquid's not going to leak out, it's going to keep the instrument sterile. So now there's no risk for contamination. Our EHS group has approved that this will be the only instrument on campus that has a microscope that is allowed to run BSL2 plus samples. So it is small. It's far smaller than a cytometer. It's probably like yay big. It's interesting. And that's where I love ChUG and I love these types of podcasts and talking to people because, like, you only know what you know. And it's right. You don't know what you don't know. And, I was completely ignorant for a lot of the spatial biology stuff. So I've really appreciated getting to hear and learn a little bit more as well. And it's almost hard job for us nowadays, I think, as core managers, directors, and stuff like that, just to be aware of what all is out there as we start making these decisions. Because when the money comes, it's usually like a knock on the door. Hey, by the way, we got $200,000. Like, go get something. You've got a week to figure it out. It's hard to make that decision.
Yeah, I always thought that if I had to rebuild my core, what it would look like, and it would not look like what I have right now. So it's good to know other options are available. Before we get to running a core in spatial biology, this is just for my own curiosity, and again, I don't do that type of stuff much. But when you do, we talked about getting multiple markers relies on cycling your staining process. So basically, you stain first, take your image, remove the antibodies. That portion of removing the antibodies, I think, is done in different ways by different companies. And I just know the one from Miltony. I'm guessing they use their releasable antibodies. Do other companies have similar type of technologies? And ultimately, do you have a wide range of clones available to you to run your experiments, or you're kind of limited by that specific technology that you need to use to release the markers?
So kind of yes and no about the clones, right?
Mm-hmm.
So like I'm coming from personal experience with Codex system, right? Like you can get about, I don't know, maybe 50, 60 antibodies directly conjugated. And those antibodies are conjugated to DNA barcodes, right?
Oh, right.
You can buy this directly from Akoya off the shelf, right? And they will ship in a few days and you will have it here. They are optimized in a way, right? So you can start using them almost right away. Well, you have to probably run some tests pilot to make sure that the tissue sample that you want to look at actually works well with this set of antibodies and so on. But that's one source, right? Akoya also sells the conjugation kit that you can get from them and then try and conjugate any antibody that you want to include in that panel. So it's always a combination. Some groups that I work with, they prefer to conjugate a lot of antibodies themselves because they already coming from, a large library of antibodies that they tested on the particular tissue, right? So they have validated over the years, dozens and sometimes hundreds of different clones of antibodies, and they know what works the best for them and for their tissue. But they conjugate, basically. Doesn't seem to be very difficult, and that's what they run. In terms of Codex, right, the advantage is that you stain your sample with all the antibodies at the very beginning, right? So there's no staining after that, and then you cycle the barcodes, right, with fluorescence. The case like with Maxima from Miltony, right, or from the case of the Luna4 company that they have the instrument called Comet, right? There you cycle your primary antibody and you come with the secondary antibody, and it's all done by the instrument. You have to optimize your assay, a little bit more optimization, but you can use any antibody you want, right? So you're not really limited by what's already barcoded, right? Conjugated barcodes, or that you would have to barcode it yourself. So then the pool of the different antibodies is much, much larger.
Okay.
Again, right?
You can also get creative if you want to get away from antibodies. I'm not sure if you've had any experience. I haven't had any personal, but it's been a hobby. Right? Like all of these extra things, like these aptamers, these anchorions, like, the idea that we can use protein engineering to make things with high binding affinity. But then that also gives us some pretty unique options where, you can hit it with specific wavelengths and then destroy them or cause them to release or to do other things as well.
I think tinkering would be something that I would love to do. I just don't have much time. So you know what, like when I switched to core facilities, I realized that the big aspect that's extremely important right now to me, and it wasn't important that much before, is to have something that work well, and I don't have to spend months and years actually, getting it to work. So like when I love,
where's the job security in that?
So I do love idea of changing and looking at different chemistry, right, and exploring different reagents. It takes so much time, right, and troubleshooting and optimization that I don't find it possible right now, at least for me in my setting, right? So like if I have the chemistry that works and I can, change it maybe a little bit here and there to optimize it, change the concentrations, change the exposure time, maybe conjugate a bunch of antibodies myself, I'm good to go, right? And so then I would probably choose that path right now. But there's so much going on in the field, right, that's not commercially available. So you go to original paper and you see what people are actually doing. Unfortunately, I'm not one of those people right now. I would love to maybe in the future, right? But like there's so many different approaches, branching chemistry, right? Combinatory type of approach when you can detect, using fluorescence, detect thousands of different, molecules and so on. Really cool stuff. It just I need a little bit more time on my hands and maybe some money.
We're in a unique space at MSU, so we don't have an immunology department, and so with our flow core and everything else, we're dealing with like everything else, that isn't immunology. And the immunology that we do have is not in human, mice, or rats. It's, tigers or flamingos or all of these random things. And so I love it for that because somebody could bring to you like, okay, I got a simple project, it's T cells, B cells. And you're just like, what's the catch? You know what I mean? And they're like, you know, it's in this species that, you know, nobody has ever seen or characterized before, you know, and it's like, where do we even start? And so like we've been using these like alternative ones a lot more lately, and they work really well. But I think that's what's holding us back from diving more into the spatial biology as a core, because so many of the platforms are locked down that it's like we can do it right now with a high content and figure out creative approaches as the field continues to evolve to that point. But I'm at the same time incredibly jealous because you get to see all these other cool toys. So
Oh, but that's really cool, you know, like flamingo. I would love to, you know, look at the immune system of flamingos and what kind of color of shrimps they eat and how does it affect B cell and T cell ratio, for example. Here's the project.
right?
But I think Cezary's point is a good segue into the last thing I wanted to talk about, which is like how to set up a spatial cytometry service. And for the purpose of this discussion, we'll kind of skip on the confocal cycling disk stuff, because that seems to be something that can be included in different places. But like it seems to me that these high content platform have their own set of requirements for the operator, for the person running the and you can prove me wrong. Like I'm just talking on top of my head.
You're bored with it already. I get it. Those
No,
are like, we can do hundreds, we can do tens of thousands of things. You're bored with my 4.
So I have this CytoU presentation coming up, and it's about marketing your technologies to your users, and how to basically develop a whole framework where what you're going to propose will actually be useful, so that when you have to report to whoever gave you the money to buy the machine, you'll have something to tell them that's positive. Coming up, anyway. But how do you do that with spatial biology? And I'm in a framework where you know, University of Chicago, we're all silos. You have like the flow stometry, microscopy, and everybody is that their own little turf. We don't really talk to each other much. And I guess
They're just all intimidated by your beard.
No, that was before. The beard came after. The ultimate best response, obviously, is probably to say that yeah, you need a collaboration between cores, and it's very loose structures. But let's imagine that you're running it at University of Chicago, and then you're going to be the spatial biology person. Like how would you so first off, like how big a market do you think that is? How many people are interested, and what are the kind of competition to that service? That like competition would just mean people trying to get an answer. They could do it by spatial biology, but they might prefer another technology with which they're more familiar with, say. So we're just talking about the context of that technology, like who's in charge, who wants to use it, what else can they do if they don't want to use spatial?
Maybe I'll start, because that's the kind of problems I've been trying to solve from the beginning, right? Like, I think it's difficult, at least the spatial biology part that I'm doing right now. I think it's difficult because it's so new. So it's like there are people that are not even aware it exists, right? And they do research, you know, multi-parameter flow cytometry. And then you have to kind of tell them, you know, like, we can also do that. And like, you know, the selling point for me is, that's what I already said, right? Like the logistics in terms of getting the sample, treating the sample or not treating the sample in this case, right? So that's easier. That's the benefit. Second benefit would be it gives you the extra layer of the information. So it gives you the spatial context, but that we don't have with flow cytometry, that we destroy by, you know, just performing the dissociation. So there's a new layer of information that's unexplored, really, for decades, how those different cells interact with each other and how they organize and form the tissue, you know, and then Then you can include the aspect of the disease and versus healthy, versus solid tumors and infiltration and all this. So this is a new field that we just created and we have a label, right? But again, because it's so new and the tools are emerging and tools are coming in, so it's very difficult for new people to jump into that because it's all unexplored and there's no standards, right? So like we kind of right now, we are creating the standards and we will see in the future, in a couple of years probably, which technology will get there. But like also, I also like to think about the workflow a little bit beside the instrument itself, right? So I had to learn how do we prepare samples, right? Because I had to learn how do we prepare the samples, because without the instruments wouldn't be very useful. But then also I had to learn what do we do with the data after that, right? Like we have FlowJo, we have FCS Express, we have Omic that already set up and ready to go and relatively easy to learn and enter, right? For me, like I spent a lot of time working on the data processing, right? Like you have, like from the platform I'm running, what I got at the end is like thousands and tens of the thousands of individual images, right? What do we do with that stuff, right? Yeah. So I had to find a way and use something that is useful, right? So like now we can create one single image, like really large single image. Sometimes those images are 10 or 15 millimeters by 15 millimeters. It's a big chunk of tissue or multiple different pieces of tissue on the same slide, right? That we can image. And then we had to find a way, put it together in one format, and then have all the data at the same time. So what I can give you right now, after months and months of troubleshooting and coming up with different ideas and testing those different ideas, this is a data processing pipeline that I have. And I give you a 40 color large format image that you can look at, right? But then after that, there's analysis. So that's the data processing, and like, what do we do with that data? You have a beautiful image, but what does it mean, right? So then I had to kind of think, how do we identify cells? That's the segmentation, right? Like, how do we quantify the expression levels, right? The emission versus the expression levels here, and then like, how do we quantify interaction between different cells? All those, the analysis part, like I won't be able to really do, right? I have some ideas and I have pointers for people that I'm working with, but like really it's on another level. So like it's very hard to support the analysis without proper training, proper people, right? Like that's something very difficult. But I think by providing solution to the bottlenecks that I just mentioned, I think we have a better chance to adopt this technology across the scientific universe a little bit faster, a little bit easier, a little bit better, right? So that's my way to approach this. I don't think it may not be the best way, but that's how I think about it.
Mm-hmm. I loved a few of the points that you brought up. I think a lot of people focus on knowing how the instrument works, but really that does not help you to grow your core, I think, as nearly as much as knowing how the assays work and the sample prep works, which I think is so crucial and fundamental, but it's such a bigger challenge. But that's also why I think that we all are successful, is again because of the community. This flow community is unlike anything else, right? I can remember before becoming a professor and in grad school and the rest swearing off academia, and I would never do this because of the competitive nature, and like, was meeting people like Dave when I got into flow, where it was just like mi familia, like all the same people that just wanted to help each other out, and that's how we learned. We were able to perfect these assays. The other thing that struck me in your conversation, I think that we're all going to start thinking about too, is hiring biostatisticians, bioinformatics folks. I mean, the problem is you already talk about the gap between academia and industry pay, and like, I mean, that's like on a whole nother scale there. But like, it's going to be something like that that's going to tie all these fields together, because as much work as it used to be in flow, where all the work was in the assay development, once you did the analysis, it's like, all right, draw 3 or 4 gates. It's not that hard. A little more complicated when we started getting into like dimensional reduction and cluster analysis, but again, when you start throwing in the sheer amount of data and trying to come up with relations in the data that you can get from, millions of images and the morphological parameters that you can derive, the spatial information you can derive. I mean, that's like, I think that becomes 90% of your workflow. And it's hard to just give that to a graduate student with a few tools and say, like, have fun. I mean, you need that for it to be successful, but like at some point, and it's not nearly as automated to do all that stuff. Maybe it will be within the next 5 years, but like being able to have almost another like analysis core individual be able to do that and work with people will be huge. The other big bottleneck that I can think about this comes back to how we expanded our core. Because again, no immunology department, like again, go back, anybody watching this too, think about your billings and how much of that's related to immunology. Like how do you keep a core open without immunology? Was just going into every other group and every other lab and just saying, and out of general curiosity, like, what do you do? Right. And how can you help? That also helped us to bridge relationships with the cores when you have silos, like you mentioned, by saying, you know what, I can't help you with that. But I was learning more about mass spec the other day, and they just got a new instrument that seems like it'd be right up your alley. And we started doing those things, which then opened the doors to meeting those folks, which led to me going to those cores and being like, what do you guys do? What are your instruments? How do they work? Them wanting to know the same thing back, that we all started working together to help each other out, which again was a great feeling when you can break down some of those institutional walls and realize that it's not just flow cytometry that's great people. Like, there's tons of great people in all kinds of fields. But going into those new groups, you'll have the new PIs, I think, that come in that already are aware of it and are happy to spend the money. They have their startup funds. They know what they're getting themselves into. It's harder for somebody that's already doing something, who already has their grant and their money planned out, to do this. And so we grew cytometry in that aspect by being able to get free reagents, free instrument time, and helping them to design the assay, such that we could say, look, the next time that you got some spare cells, let's have them. We'll add a few colors, dyes, things in. We'll run it, we'll click, and look, hey, look, this looks like it would work for you. Boom. They've got a few figures at least that they can take to go get the grant money that they can get excited about to start branching things out. But again, it's easy to do that with a flow reagent that costs a few $100 and instrument time that's only 30. It's a lot harder when the entry fee is $5,000.
Yeah.
And I don't know how to apply that same thing, because like for so many people doing something new, seeing is believing. And if it's not either new faculty that have that, where then you can point to them and say, like, look, if you can get the money to do this, like, look at how great it can be for you. When everybody's response is, well, yeah, like if I had the money, I'd love to do that. And I don't know how to break down that hurdle other than trying to do it in alternative ways. If you can't have the money for the whole spatial biology thing, like these anchorions and all of these other recombinant proteins are significantly cheaper than antibodies, and they're all growing markets. And so we've had a lot of success reaching out to them for samples, where that is, like maybe we can get you 5 or 6 to start with. And, we can start branching out to the things that are free, like the analysis, trying to see if there are spatial relations, trying to get that preliminary data to then want to go deeper, and trying to figure out how to make that free and easy to use. And I think that that's where, again, we go back to the community and thinking, like, what are those things? How do we get the most bang for our buck to make some true believers?
Yeah.
Good points.
I think there's some technologies that are easier to introduce, like spectral flow was easy because it's mostly flow cytometry, but more like bigger, so that people kind of very quickly caught up. And but with spatial biology, it feels to me that there's value for a core facility to actually buy the instrument and kind of sit on it and just explore what can be done, and then come up with different experimental pathway, and just don't pressure that core to make their ends meet just yet. Just at some point it's going to pay off. But
I mean, it'd be really cool if companies did something like an extended demo with one of these instruments, where it's almost like you were a beta site for a year.
Yeah.
So like I don't have to get recharge on it, but I can just get people in and be able to use it. There was another instrument that we demoed recently, and I think that it would have had a great place in our core, but the reagents they couldn't even provide the reagents for free for the demo, and the reagents cost a couple of grand. And so it went from like 20 people that had interest down to 3 that
Right.
had the money to afford the reagents. And it was enough reagents to do like 100 different assays, and like they just wanted to test it with one, couldn't use it, couldn't return it. And it was like, yeah, I could see how this would work, but it would require more optimization. Nobody wants to do that for an instrument that you're not committed to buying, committing to using, that's only there for one day. It's hard to get a real sense of these things. Which is why if I would have won the $1 billion lottery or whatever that was a few weeks ago, I would have loved to have like buy a bus and like put a whole bunch of cytometers and instruments in there and just like take it on the road, right? And just like pull up this, Michigan, we got tons of smaller universities where it's just like roll up for a week and like, hey, people, who wants to do some cool science,
Right.
and just introducing people to these different things, which is great.
It's a good point. I mean, we're all networked nowadays. You should be able to like set a demo instrument somewhere and keep a whole bunch of people aware of what's going on in that place. And like you said it at University of Chicago, say, and we'll broadcast and we'll tell people what's going on, and people can ask questions, stuff like that. And companies would benefit from that. It feels like a good idea.
I think that that's going to happen more instead of them trying to go to the giant national conferences. Because again, the dynamic has changed, right? The regional conferences are picking up. This is a good plug for GLIFCA. So both Dave and I are involved in our regional flow cytometry conference, GLIFCA. And we really kind of took this to heart. And this year, we're going to have instrument demos at the conference for a day, with the idea that as a core director, like I know the instrument works. I feel like a lot of times they do a demo, it's trying to prove to the PIs that the instrument works no matter what your sample prep looks like. But for us, it's just like, I just want to see what's out there. I want to click the buttons and see things. And so ours is going to be a demo for a day, and as well providing opportunities then and making it cheap that small companies that have new things, exciting things, can bring it in. We can see what's going on and get excited about it and get some hands-on time. And I really feel like a lot of other conferences are going to start shifting to the same format. Nobody's got time to go to the 1 a-year international conference, but like you said, oh, hey, Chicago is going to have a couple of different instruments this year. Like, yeah, I'll go find time to hop over there for an hour or two and to check this stuff out and to learn, if not just because you want to go over to see Dave.
Yeah, come and see us. Don't worry too much about the deep dish pizza, but the rest of the food is pretty awesome.
Right on.
Yeah. All right, guys. Any other things you'd like to cover? I ran out of ideas.
I'm just excited about the field, right? We've got like 5 different imaging cytometers coming out. So like,
yeah,
imaging is going to be big. And again, it'll be a few years before I think, everybody's fully on board with a lot of the spatial biology. It's exciting to be at the point again where it feels like the beginning of spectral, where it's like, oh man, there's so much potential and ideas going around. And that's really what it feels like this whole spatial biology is at right now. I'm just really excited to see where it's going.
I agree, but very exciting field to be in, and I think it's bigger than spectral flow cytometry that we experience because it's so new, and again that different layer of information that we are able to look at and kind of understand and quantify. It will take a few years, and I think we will see a few different instruments appearing on the market and falling off of that market, I guess. And then the cost will be a little bit more clear in a few years, I'm guessing. And, with the leaders staying up and running everything a little bit better and faster maybe. But it's a great field to be involved in, and we'll see what the future will bring.
Yeah, sounds good. Cezary, you're the only instrument on campus right now, right?
No, I have one that's the spatial proteomics, but next door there is the GeoMax, which is the spatial transcriptomics that you pick your own area in a way, right? From NanoString.
Okay.
And so that's the sequencing based.
That's the sequencing core?
Yeah, that's sequencing core. So they have one. Yeah. And I think that's all within the core facilities, but I know there's few different labs that have few different instruments. I know there is Visium from Tanex in one lab here. I know Cosmix there's in one lab, and there's a couple different codecs or iteration of different codecs, with different configurations. So like that's what I'm aware of.
Okay, sounds good.
Yeah. So yeah, it's definitely taking off slowly but surely.
Yeah. All right, guys. Thanks for your time. Thanks for viewers for following us, and we'll talk to you later.
All right. Thank you, guys. It was fun.